Hi everybody and welcome back. Like we've been doing for the last couple of labs, I wanted to give you a very brief overview of the Mesozoic life that you'll be encountering as you work through Lab 12. So let's jump right in. So looking at the Mesozoic era, we're going to start with the early Triassic and mention a little bit about the Jurassic and what's going on then in the Cretaceous. So we pick up the Mesozoic right where we leave off the Paleozoic. Pangea is still fully assembled. The planet is still pretty dry, kind of cool. The temperatures start to warm up a little bit in the early Triassic, and this is when we get our initial first stages of rifting. So Pangea forms throughout the Paleozoic, and around the middle to late Triassic, it starts beginning to rift apart. But it's not all at once. It's a couple continents separating here and there. So when we look at a map such as this of the early Triassic paleogeography, you notice that, again, Pangea is firmly connected. And then the darker browns that you're seeing there, those are all areas where we have mountain ranges. And the big one between North America and the main part of Pangea, South America and Africa, we have the Trans-Pangean Mountains or the Appalachians or Allegheny orogeny. So that's Himalayan-sized mountains. This causes that rain shadow effect that we saw in the Permian, and it continues into the Triassic. So again, this is a good opportunity to remind you that changes in periods, changes in eras, going from Paleozoic to Mesozoic, is not necessarily a change in climate or geography, it's a change in life. So what separates the Permian from the Mesozoic, or the Permian from the Triassic, is that we lose most of our Permian life. Landscape-wise, though, it looks very, very similar. So the Triassic is a continuation of the climate conditions that we left off in the Permian. So we start out with the early Triassic. This is about 237 million years ago. And this is what the world looks like at the end of the Cretaceous, or approaching the end of the Cretaceous, around 94 million years ago. This isn't even the end of the Cretaceous. This is what we would say is like the middle to late. And notice that, again, this doesn't look like our modern globe today, but it's certainly getting a lot more familiar. Antarctica is down at the South Pole. We do have rifting and separation and a proto-ocean between South America and Africa. We have a central Atlantic, and we have a Pacific Ocean. The Tethys is still there, but it's getting smaller. And notice that most of Asia and Europe are assembled, but we have some changes in sea levels. India is still hanging out in the southern hemisphere, things like that. And by the time we get to this time period, temperatures have gone from dry and cool in the Triassic to hot, very hot, and humid in the Cretaceous. So the Jurassic period in between is this cool, kind of neat transition going from dry, cool, arid to warm, sweltering humidity of the late Cretaceous. We've been looking at those slough sequences, kind of following along with what's going on with sea level throughout the Phanerozoic. So we talked about the Sauk sequence and the Tippecanoe sequence in the early Paleozoic, sea level rise and fall, rise and fall. And then the Kaskaskia and the Absaroka in the middle to late Paleozoic. Now the Absaroka is a really big sequence, right? It spans from the early Mississippian through at least the end of the Permian, a bit into the Triassic. And that's because half of this is actual marine sea level. The other half of it is sediment that's been accumulated from deserts. So that's what explains what's going on with the Absaroka sequence. We have this huge unconformity afterward, and then we have the Zuni and the Tejas sequences in the Cretaceous, then in the Cenozoic. So what we're looking at throughout the Mesozoic is a time of rising sea levels, ultimately ending with a very brief fall. So that's what we're going to be looking at, is what's being deposited in that Zuni sequence. So again, we're going to be looking at a lot of the kingdoms and phyla that we first introduced in the Paleozoic and kind of see what they're doing in the Mesozoic. So starting out with Kingdom Animalia, which is we'll look only at animals today. We're going to revisit phylum Cnidaria, class Anthozoa. Last time we looked at this, we looked at the two Paleozoic orders of corals. That was the tabulates and the rugosans. Those are all gone now. They're completely extinct. The order Scleractinia appears then in the Triassic period, late Triassic. It really diversifies in the Jurassic and are still around today. When you go snorkeling or scuba diving, you see things like Archaeohelia, which you can see in the black and white image, and brain corals and staghorn and elkhorn corals. All modern corals are Scleractinians. However, they do have some similarities to what we see with our rugose corals from the Paleozoic. They both have radiating septa. The difference is how those septa form. So if we look at what's going on with the septa of rugose corals, they grow these main septa towards the center, and then next after that, another set grows into the side, and then another set grows into the side of the previously grown septa. And that's what we see in rugose corals. We call that serial insertion. And you end up with that kind of spider web looking pattern of those radiating walls. With scleractinians, they all grow towards the center. Sometimes they meet, sometimes they don't, but they don't intersect with each other. This is called radial insertion. So it's not really important that you know the difference between radial and serial insertion, but it does help to know the difference between the types, the general differences, that they have different types of septa. They both have them, but they're a little bit different. So all of our modern corals are scleractinians, but they do have some similarities with rugose corals. Possibly an evolutionary lineage, we're not really sure. Our next phylum is Mollusca. Again, we've looked at mollusks in the past, especially things like cephalopods, but we're going to be focusing on bivalves today. So remember, at the end of the Paleozoic, we lose most of our brachiopods, which were the two-shelled, filter-feeding animals that were living in the marine world. However, in the Mesozoic, starting in the Triassic, mostly in the Jurassic, is when we get our diversification of bivalves. Now, bivalves are things like clams and oysters and mussels. They've been around since the Cambrian, but they've always been a minor group behind brachiopods. But now that most of our brachiopods are extinct, bivalves fill that niche and diversify. And they start out kind of weird. There's a couple of genera you might see in the lab. One is Venus, which is just kind of your typical clamshell. Exogyra is this really big guy that we have on the screen here. It is a coiled growing oyster, but their shells are like 2 to 3 inches thick. And that's because things like big marine reptiles from the Mesozoic and the Cretaceous were swimming around eating these things. So they're really heavy, thick oysters compared to our modern day oysters, which are pretty thin and kind of irregularly shaped. Any bivalve, whether it's an oyster, whether it's Exogyra or Venus or a pecten clam, doesn't matter, all have some similarities. And you can see that in the top image on the screen, kind of the anatomy of a bivalve. If you look at the inside of a bivalve, you'll see two circular structures called muscle scars, and these are what help keep that shell closed when they want to close it. There's muscles that hold the two valves together that close them when they flex. There's also this thing called the palleal line and palleal sinus. That's an outline on the inside of the shell that shows where the actual soft tissue fleshy animal was adhered to the inside of their shell. So they are a mollusk. They're related to things like snails and octopus, but they're their own class, bivalves. When you're walking along beaches today and you pick up shells, you're usually picking up bivalve shells. So they start out really, really weird, and then by the time we get to the Cenozoic, a lot of our bivalves are a little bit more familiar to what we see today. We still have cephalopods in the Mesozoic. In fact, in the Mesozoic, cephalopods go insane in all these different varieties and forms. We talked about nautiloids in the Paleozoic, and we still have 1 or 2 species of them alive today. But this one particular order of cephalopods, the ammonoids, or Ammonoidea, really diversified like crazy. Ammonoids first appear in the Middle Paleozoic, but they're very, very minor, and it's not until the Jurassic, especially in the Cretaceous, they go insane with their different forms. You can see some of the different shapes and sizes we have of ammonites here. So what makes an ammonite different than a nautiloid? A lot of it comes to the structures on the shell. So for example, you notice that on nautiloid shells, we have the sutures are very smooth. And that's because, remember, the sutures are what's locking one chamber to the next. When we start looking at our different suture patterns of our ammonites, we see a variety of this. It goes from being a straight to slightly curved line to what we call the goniotitic sutures here. Those are rather simple ups and downs, sometimes a little bit sharper. And then in the Jurassic into the Triassic, we start seeing more of these cerotitic, which have loops and then some little wrinkles and loops and little wrinkles. And then we get to the ammonitic ammonite sutures. And that's what you see actually in this photo here. It looks like a fractal pattern. That's how these shells are interlocking with each other. Okay, and what that's helping is ammonites are so diverse in their shapes, they're actually living in deep water, shallow water, middle depth waters, all these different environments, and they can move around them as they want. if you're going to go down to great depths, you need that structural integrity to keep those shells from imploding. So it keeps the shell nice and stiff, and it keeps them from really being pulled apart too easily. So nautiloids do this still today, but they're not doing the same kind of stuff that ammonitic ammonites were doing. So the sutures, the bonding between the chambers becomes really, really complex in ammonoids. And actually, it's one of the ways that we use to identify ammonoids, different ammonite species, is what's going on with their suture patterns. Another cephalopod that diversifies in the Mesozoic are belemnites, order Belemnitia. Belemnites are only about this big. Okay, they were a small squid-like animal, but what makes them unique is that they actually have no external shell like an ammonite or a nautilus. Belemnites have a hard part called a phragmacone on the inside of their bodies, as you can see in this diagram. It's a little calcium carbonate bullet-shaped fossil. They're about this big, and actually that's what belemnite means. In Latin is bullet stone. They look kind of like a 50 caliber round. So when these things died, we'd find literally thousands of them because they would swim in these cute little schools of thousands and thousands of belemnites, and then they die, they decompose, and the only thing left is that internal hard part. So unlike an ammonite where you have a shell the organism was living in, this is a hard part that the organism lived around. So it was that little bit of an internal skeleton. Very similar to a modern day cephalopod called the spirula, which has a small coiled internal structure, but it's not as heavily formed as belemnites. And then we get to dinosaurs. Okay, so we're going to talk about phylum Chordata, subphylum Vertebrata, class Diapsida, suborder Dinosauria. So just to walk you through, chordates are anything that has a notochord, so a flexible backbone. This would include things like eels and lamprey. It also includes vertebrates, which are animals that have a vertebrae. They have a vertebral column and a skeleton. Within vertebrates, we have a number of classes. One of them are diapsids, which we're going to talk more about. That includes snakes, reptiles, birds, crocodiles, and dinosaurs. And then superorder Dinosauria. And dinosaurs are pretty diverse. As you can see, we have two main groups, the Saurischeans and the Ornithischians, and this has to do with the way that dinosaur pelvises are assembled. And if we go over to the Saurischean side, that's where we see the meat-eating dinosaurs, what we call theropods. It also includes modern-day birds. You can see how they branch off of that. And then we also have the sauropods, your long-necked dinosaurs. Both of those are Saurischean. On the Ornithischian side, we have all of our plant eaters like the ceratopsians, the horned dinosaurs, the thick-skulled pachycephalosaurs, the duck-billed dinosaurs, the stegosaurs, all of those. Together, these all make up Dinosauria. What I really want you to know, though, for the lab today is some major parts of the dinosaur skull. Okay, and I'm going to be using an example that I have here at home, and also you'll see it in the lab as well, in the lab manual and as you go through the lab exercise. Okay. So here's a diagram of an Allosaurus skull, which was a Jurassic predatory dinosaur. If you remember walking into the main lobby or first hallway in Harrington Hall, this is the skull that's in the big display case. Most meat-eating dinosaurs have very similar skulls when it comes to what types of shapes and structures and bones that they have. It's not just one bone in the upper skull and one bone for the lower jaw. Reptiles have lots and lots of little bones, and also extra openings. So we're going to follow along on this diagram, and you can follow along in your lab manual as well. It's on the very last page of the lab manual is this diagram. And also I have this, one of the things I've been doing in my free time here at home is 3D printing things. And one of the things my son and I 3D printed is a life-size velociraptor skull, which was kind of fun to do. So just to kind of follow along again with your diagram, the very, very front of the skull here is called the premaxilla. And if you look carefully, you can see there's a bit of a line that goes down here that separates the premaxilla from the rest of the skull. This is really the snout. We don't have a premaxilla, but a lot of animals do. It's a separate bone from the rest of the tooth-bearing elements, is where the very, very front teeth are. Behind that is the actual maxilla, the main tooth-bearing bone of the skull, and you can see it on the Allosaurus on the screen as well. On top of those, you have nasals, right? The nasals run on top of the skull. We have nasals too. We have a maxilla, our upper tooth-bearing bone, and we have nasals. So when somebody says they broke their nose, they usually didn't break their nose. They broke maybe the cartilage in your nose. That's what really gives your nose its shape. But if you feel right here at the bridge, like if you're wearing glasses, where the glasses would rest, that bony part right at the top of your actual opening for your nose is called your nasals. Now, our nasals are about maybe an inch long. On Velociraptor, they're about 6 inches long, maybe 5. On T-Rex, they're about 2.5 feet long. So they really kind of vary, but this makes up the top part of the snout of the skull. We have it too. The bone right in front of the eye is called the lacrimal. It's this thin bony bar here for Velociraptor, and you can see it's kind of a T-shaped bone between spots 2 and 3 on your screen for the Allosaurus. The lacrimal is the bone in front of the eye. Okay, we have one of these as well. It's right here on the inside of your eye, right by where your like tear duct is. There's a little bump. That's your lacrimal. Okay. Post-orbital. So here on our Velociraptor, if this was the lacrimal, this is the post-orbital bone, and you can see it again between 3 and 4 on the diagram on the screen. The post-orbital, as the name implies, post meaning behind, orbital, the orbit, where the eyeball goes. We also have post-orbitals. Please don't touch them. They're behind your eyeballs, and that's gross. Moving along, the very top of the back of the skull we call the parietals. Again, we have parietals. That's your 2 bones here at the top of your head. It also has a frontal. Frontals are your forehead. We have that as well. And then squamosals here on the back of the skull. Okay, the squamosals run on the back. That's kind of this bare area that you have at the very, very back of your skull. Okay. Quadrate, that's going to be this bone down here that articulates with the lower jaw. So the lower jaw here can be removed. And we'll talk more about that in a minute. The quadrate is the bone back here that the lower jaw attaches to your skull. Okay. There's also a couple other little bones here. You think about it, we as mammals, we have 1 bone making up our lower jaw. Reptiles actually have 4. They have the dentary, which is the lower jaw that has teeth in it. And then they have all this part back here. That is the cerangular, angular, and articular. And I'm not going to hold you to memorizing all those names necessarily, but there's 3 extra bones in the lower jaw. Okay, and this is true for all reptiles, whether it's a bird, a crocodile, or a dinosaur. So why do mammals only have 1? Well, actually, we still have 4. We still have a lower jaw. That's really the dentary. Our mandible is really the dentary in a reptile. But what about those other 3 bones in the back? Well, think about where your jaw articulates to your skull. It's pretty much here by your ear. And the 3 smallest bones in your body are in your ear, and they help with hearing. And that's how reptiles hear, is with their lower jaws. So those 3 extra bones, what in reptiles we still call the articular, cerangular, and angular, are still present in a different form in mammals, such as humans, the 3 tiny bones in your ear. That's where they come from. We can trace that embryologically. And then finally, the jugal is your cheekbone. And on this dinosaur, it's on the Allosaurus on the screen, it's that lower jaw under the eye. And on Velociraptor, that's right here. That's the jugal. Okay. So we have more than just bones, though. We also want to talk about openings, because that's what makes this a diapsid. Okay. The first hole we're going to look at is the nares, not to be confused with the nasals. The nasals are bones. The nares is the opening for the nose. It's your nose hole. We have a nares, an external nares as well, or a nares. Okay. I'm going to skip 2 real quick and we're going to talk about 3. 3 is the orbit. Okay, that's where your eyeball goes. Okay, so if I focus Velociraptor right at the camera, okay, right here and here where my index fingers are, those are the orbits. So this guy could look right at you. Okay, it had forward-facing orbits or forward-facing eyes. I just had to look down its snout. Okay. Now, why did I skip 2? Because 2 is an opening in dinosaur and a few other extinct animal skulls. It's called the antorbital fenestra. And if we break down that word, ant as anterior or in front of, orbital, meaning the orbit or eyeball hole, fenestra, which is Latin for window. So it's the window in front of the eye. Okay, it's number 2 on the diagram. So why do dinosaurs have an antorbital fenestra? Simply, it lightened up the skull. They evolved these bony struts of the lacrimal and the postorbital, so they didn't have a lot of extra weight, and so they have this opening. Now, it would have been covered with skin. Some animals still have these today, but the antorbital fenestra is that kind of forwardmost large opening, and it was a place to lighten the skull. It took out some weight. Okay. Now, 4 and 5, which is at the very top, these are really important because these are what separate diapsid reptiles from synapsids, or ultimately mammals. On the lower part of the skull, we have this extra opening. Okay, so it's behind the orbit. So here's the orbit. We're talking about this opening back here. Okay, number 4 on your diagram. That is known as the lateral temporal fenestra. So lateral, meaning lateral, it's on the side. Temporal, in the temporal region. Fenestra, opening. Okay. Why do we bring this up? Because mammals and dinosaurs and other reptiles have this. This is a common characteristic in most amniote vertebrates. We have it. We actually do. So here's your cheek. If you go and trace the bony arch from your cheek all the way to your ear, okay, that's called your zygomatic arch. Above that, you can feel your temples, and if you move your jaw, you can feel the muscles moving. You can also feel them below that. So if you actually look at how muscles attach onto a human skeleton, that bar, your arch here, is allowing muscle attachment to your skull. And that's exactly what this is for. The lateral temporal fenestra is a place for muscles to kind of wrap around. It gives it a stronger bite force. Except diapsids have an extra one, and that is the supratemporal fenestra. Supra, not super, but supra meaning above. The above temporal window, so the window above the temple. And to really see this, we have to turn our skull and look right at the top. These extra holes here, extra muscle attachments. This is something all reptiles have. Crocodiles have this. Dinosaurs have this. Birds have this. So they have a place to wrap muscles around the side and up in top. That gives them a super duper bite force. Mammals, humans, we don't have this. We only have the lateral temporal fenestra. We don't have a supratemporal fenestra. That's a diapsid characteristic. That's what makes it a diapsid. It has 2 extra openings. Okay. So when we think about how these features fit together, okay, this is what increases that bite force for something like Allosaurus or T-Rex, or surprisingly even little Velociraptor, which, yes, by the way, this is a life-size Velociraptor skull. They were not very big. Sorry to ruin Jurassic Park and Jurassic World for you, but Velociraptor was only about 6 feet long and about 3 feet tall, and its head was only about 9 or 10 inches long. It wasn't the giant things that you see in the movies. But this is why they have a very, very strong bite force. So like I don't want to get bit by a dog, but if I had to choose between a dog or a crocodile, I'm going to choose the dog because while they have a lot of muscles, it's not like a crocodile, which have those extra bars in the back of their skull to really clamp down on things. Okay. So now go ahead and, with that information under your belt, go ahead and get started with Lab 12, Mesozoic Life. First, make sure, like before, you read through the assigned readings and complete the pre-lab. That's on Canvas. Make sure you do both of those before you get started on the lab itself. And also make sure you're looking back at Appendix A, which I posted on Canvas, which will also help you with some of the biology of these organisms. Good luck.